Gregor Mendel serves as the historical architect of predictable trait inheritance. This botanical illustration of the garden pea plant demonstrates why it was the ideal controlled biological model. The distinct physical variations of its seeds and flowers allowed Mendel to observe strict mathematical patterns across generations. To understand those macroscopic patterns, we must look past the physical plant and drop down to the cellular level, examining the molecular hardware of heredity. That hardware is built on the gene. A gene is a highly specific, functional segment of a DNA strand that determines a particular trait. The exact physical address of that gene on a chromosome is called its locus. This coordinate is fixed. However, the genetic sequence located at that address can vary. We call these alternative variations alleles. For example, a single locus controlling plant height could hold an allele for tallness or an allele for dwarfism. Because organisms inherit two alleles for each trait, one from each parent, they can pair up differently. An organism carrying two identical alleles at a locus is homozygous, while one carrying mismatched alleles is heterozygous. This creates a strict division between an organism's genotype, the hidden molecular code it carries, and its phenotype, which is the observable physical characteristic that code produces. When two different alleles sit side by side in a heterozygous state, they do not carry equal weight. A dominant allele will completely mask the expression of a recessive one. The recessive trait only appears if the dominant allele is entirely absent. The core of this logic is that physical traits are not random. They follow predictable ratios across generations. To understand why, we must look at the specific cellular mechanics that make these ratios inevitable. The simplest way to test this predictability is through a monohybrid cross, a controlled mating that tracks the inheritance of exactly one specific trait. If we cross two tall pea plants that are both strictly heterozygous, their genotype is TT. Each parent carries one dominant allele for tallness and one recessive allele for dwarfism. Before these plants can reproduce, they must form gametes through the cellular division process known as meiosis. During anaphase 1 of meiosis, the cell forcefully pulls the paired homologous chromosomes apart, dragging them to opposite sides. This physical action is Mendel's law of segregation. The paired alleles must separate, ensuring that each resulting gamete receives only one copy of the trait. We can map this cellular separation onto a 2x2 probability matrix, the Punnett square. The segregated alleles from each parent align along the horizontal and vertical axes. By cross-referencing the axes, we calculate the 4 possible fertilization events, generating the complete set of offspring combinations. Sorting these results reveals a strict genotypic ratio. We get one homozygous dominant, two heterozygous, and one homozygous recessive combination, a 1:2 to 1 distribution of the genetic code. Applying the rule of dominance to these codes translates them into their physical phenotypes. The dominant T masks the recessive T in the heterozygotes, resulting in 3 tall plants for every 1 dwarf plant. The classic 3:1 Mendelian ratio is the direct, unavoidable mathematical output of chromosomal segregation during anaphase 1. Biology rarely operates one trait at a time. To analyze simultaneous inheritance, we use a dihybrid cross, tracking 2 distinct features like seed shape and seed color. The starting parameters for this cross require both parents to be heterozygous for both traits. Their genotype is RRYY. To understand how these traits interact, we look at an earlier stage of cellular division, metaphase 1. Here, multiple pairs of homologous chromosomes line up along the cellular equator. Crucially, they shuffle and lock into random, uncoordinated alignments. This creates the law of independent assortment. Because the chromosomes align randomly, the inheritance of seed shape has no mechanical influence on the inheritance of seed color. This independent alignment generates 4 unique allele combinations per parent: RY, 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 and RY. Mapping these onto a 16-square matrix allows us to track the possible outcomes. Filling the grid reveals the precise mathematical outcome of independent assortment: 9 round yellow seeds, 3 round green, 3 wrinkled yellow, and 1 wrinkled green, a 9:3 to 3:1 phenotypic ratio. This law carries a severe mechanical limitation. It functions only if the genes governing the traits are located on entirely different chromosomes, or sit so far apart on the same chromosome that they sort independently. Independent assortment exponentially increases biological diversity. Its predictive power relies entirely on the untethered movement of unlinked chromosomes. Mendel's rules of dominance provide a clean binary, but the molecular reality of inheritance often violates this simple dominant recessive model. In many organisms, we observe incomplete dominance. Here, neither allele holds true dominance over the other, forcing the heterozygous phenotype to become an intermediate blend of the two traits. This botanical drawing of the snapdragon illustrates this anomaly. Crossing a pure red flower with a pure white one yields a first generation consisting entirely of pink flowers. Plotting an F2 cross between two of these pink heterozygotes generates a familiar genetic distribution: 1 RR, 2 RR, and 1 RR. Because the heterozygous state now possesses its own distinct blended phenotype, the traditional 3-to-1 physical ratio collapses. The phenotypic ratio perfectly matches the 1-to-2-to-1 genotypic ratio. Instead of blending, some alleles exhibit codominance. In this state, both alleles are fully expressed in the organism simultaneously, sitting completely side by side. A roan cow inherits an allele for red coat color and an allele for white coat color. Rather than turning pink, the heterozygote grows distinct, unblended red and white hairs at the exact same time. Complexity scales up further with multiple alleles. While a single diploid organism is strictly limited to carrying 2 alleles at a specific locus, the wider population can circulate 3 or more alternative forms of that gene. This diagram shows the human ABO blood group system, which operates on 3 distinct alleles: IA, IB, and a lowercase I. This system demonstrates both multiple alleles and codominance working together. If a person inherits the IA and IB alleles, they isolate the codominant mechanism. Neither masks the other. The body produces both A and B antigens equally, creating the unique AB blood phenotype. In all of these cases, Mendel's core law of segregation remains intact. The homologous chromosomes separated perfectly during meiosis. It is the molecular translation of those alleles that shifted the final visible outcome. This table distills 3 modes of heterozygote expression: classic, incomplete, and codominance. Incomplete dominance forces a blended intermediate, while codominance guarantees equal expression. Injecting multiple alleles breaks these matrices, expanding baseline genetic variations across a population. Beyond pure observation, these non-Mendelian patterns provide the technical framework for several fields within biotechnology. Mapping these variations allows scientists to accurately track and manage genetic diversity within specific populations. In medicine, identifying codominant expression is the basis for treating human pathologies. Sickle cell anemia manifests when a codominant allele causes normal red blood cells to twist and collapse into rigid sickles under low oxygen stress. Furthermore, multiple allele mechanics of the ABO system dictate exact rules of compatibility, forming the unyielding foundation of global blood typing and transfusion protocols. Agriculture relies on tracking complex inheritance patterns for precision plant breeding, combining favorable dominant and codominant traits to engineer high-yield hybrid crops. Mendel's laws, and the molecular extensions discovered since, reveal that inheritance is a highly mechanical process. Understanding the movement and expression of these alleles allows for the systematic prediction of biological traits.